once again to thank Gikundiro Christine and the IT people for assisting us in these online lectures. So can we move on? So you still remember my name is Jules Bizimana. Today is the immunology course. Yeah. Oh, yeah. We cannot study the whole immunology courses in one day. Actually, I try to summarize the basic content that you people in nursing you must know in this context of immunology. That's why this is going to open you to even to read more, even to search more about immunology. Hope you understand. So by starting this course, let's now come up to understand some basic terminology that we used in the immunology course. Before we go to further slides, better to know the meaning of some keywords and this immunological or immunology terminology. Before we do anything, we have to understand what is it immunology now. Actually, immunology is a branch of medicine concerned with the all aspect of the immune system. You know what's it innate immunity? Innate immunity is the first response of the body's immune system to a harmful foreign substance. Adaptive immunity is that developed. Adaptive immunity is that develops when a person's immune system responds to a foreign substance or a microbe, such as after an infection or vaccination. Yeah, next slide. So the antibody, I think is not the first time you people to hear a word antibodies. So actually antibodies refer to a protein made by plasma cells in response to an antigen. A plasma cells is a functional lymphocyte B cell. So hope you are following me very well, okay? Now, an antigen now is defined as a substance that enters in the body and induce either humoral or cellular immune response. So antigen means it is any types of substances once enter in your body, they will now activate your immune system. We also common word hearing everyone we call immunization. What is it now? Immunization. Immunization normally it's a process by which an individual's immune system becomes fortified against an agent known an immunogen. So immunization is that process, the way individuals' bodies create or produce the resistance against the disease. Hope now this is clear somehow, okay? A cytokine now in immunology refer to a types of a protein that is made by certain immune and a non-immune cell and has an effect on the immune system. Actually, a cytokine is a protein produced within the immune cell among of them. So they have an effect to the other immune cells. So serving as an intercommunication between immune cell. Okay. Next. So referring to how blood is produced. you remember, everyone, blood is produced from bone marrow. The process by which blood are produced is called hematopoiesis. And then remember that all blood cells are produced in bone marrow, where we can see where you can see hematopoietic stem cell as a mother cells may produce two types of cell, or called lymphoid progenitor cells, myeloid progenitor cells. I want just to emphasize the offspring of lymphoid progenitor cells. When during the development, this lymphoid progenitor cell may develop and differentiate into many types of blood cells, like B lymphocytes, you can see here. So you can see again natural killer cells. These are the group of lymphocyte group. So when you go to myeloid progenitor cells, it's where you have these progenitor cells may divided and differentiate into many blood cells. We can count of them. We have a monocyte here, we have a basophil, eosinophil, neutrophil, and a platelet, even red blood cells. So we are focusing now on this inflammation, what we call white blood cells, neutrophil, eosinophil, basophil, monocyte. Also adding of this group of lymphocyte, lymphocyte B and T, then natural killer cells, dendritic cells, and the macrophage. So immunology courses emphasizing on this function of these white blood cells, except red blood cells and the platelets. I think you can now understand our main purpose of immunology. We are going to deal with the function of this group of white blood cells, either granulocytes or these non-granulocytes. Yeah, thank you. We can now move for the next slide. Now, let's now introduce this immunology. What is immunology? Actually is a branch of biomedical sciences concerned with all aspects of the immune system in all multicellular organisms. Normally, all mammals must have this kind of immune cells. It's the study of immune response. So immunology is the study of how immune system respond to any antigen. It's a broader sense meaning. So this now is the study again of a cells and the molecular event that occur after an organism encounters. So a microbes or even the other foreign macromolecules. So you see that immunology refer to a study of this, how bodies respond to an antigen, or it is study again the whole process, cellular, even the molecules that intervene in all that immune responses. Yeah, next slide. You can now imagine now what the many task of immune system. You know, you people take as our immune system is our defense mechanism. It defends your body against any kind of harmful substances. It can't be microbes or any kind of substances. So this task of immune system must recognize of an antigen, what we call immunological recognition. The presence of an infection must be detected and carried out by innate and adaptive immune system. So our body immune defense must recognize an external microbes considered as an infection agent. So this kind of recognitions, yeah, it's very important. It's the many tasks of immune system. Number two, once immune system recognize an external invader, they must turn into a functional cell for elimination of infection. It can carry out by this innate and adaptive immune system. Number three, immune cells must be regulated, what we call immune regulation. It must work under control. Immune response must be kept under control. That ability of the immune system to self-regulate. It's clear, I think so. The last fourth task is to keep memory. They have immunological memory, are unique features of adaptive immunity. So they must now keep the memory for the same antigen and for the next infection. So repeat it, keep now memory the exposure of the same antigen. Next slide. So now, there are three main ways of which the immune system may contribute to a disease. Yeah, you understand sometimes when immune response reacting to any external invader, yes, that activation of immune system may result with some sign and a symptom, because the reaction between immune cells versus to an antigen may result with some symptom which is going to be characterized, which is going to become a disease. I think if you remember what previous course called microbiology, normally this disease refer to abnormal function of a body cells, resulting with the sign and the symptom. Yeah, once immune cells are activated, may activate when fighting infection and mounting an immune response, and this result in inflammation and eventual removal of the offending pathogen. Yeah, it also results in the immune system retaining a very good memory of that pathogen, which enable the immune system to mount a rapid and even more effective response to that pathogen when it infects again. Yeah, this number two, during now immune response, may lead to inflammatory reaction or inflammation process. So this inflammation process may be characterized by signs and symptoms like a fever, nausea, and something else. Number two, the way an immune response may lead to a disease, it is in the case of immunodeficiency. When the immune system may be functioning poorly, once this is functioning poorly, which makes us less able to fight off infections, immunodeficiency can occur because of components of the immune system is missing, or because of other factors are stopping it from working properly, like a cancer drug and the HIV infection may weaken your immune system. So having now this poor immune system function may lead to be susceptible to many diseases. Number three, sometimes the immune system may overreaction, or may overworking, called hypersensitivity. Even also, some immune system may now lose some control and fight against their own body cells, what we call autoimmune reaction. So the immune system may be inappropriately active against the normal body, or against the harmless substances, Autoimmune include diseases such as rheumatoid arthritis, systemic lupus erythematosus, and even many. Allergy include conditions such as asthma, hay fever, and anaphylaxis. So this it shows you that the status of the immune system may lead to some diseases. Next slide. Now, immune system in general is broken into two parts, main system. One, it's composed by innate and the adaptive immunity. The innate, in other words, it's called nanospecific response, and adaptive, it's have another name called specific immune responses. So under nanospecific or innate immunity, it's a nanospecific protective mechanism, repel all microorganisms equally. That's why it's called nanospecific. And then adaptive immunity, this is a specific immune response. These are tailored to particular types of invader. It's just specific to a particular antigen. Cannot respond all antigen the same way. That's why it's called a specific or adaptive immunity. Yeah. So both now, even if both may work differently, but they work together. Both systems work together to fight an antigen from entering and proliferating within the body. This immune mechanism also help eliminate abnormal cells of the body that can develop into a cancer. You have to know that immune systems are not limited to infection disease. Also, it can also fight against some abnormal cells that started to develop in your body and lead to a cancer disease. Yeah. So next, for the next slide. Yeah, we can now try to describe a little bit of innate immunity, called again nanospecific immune response. Under innate, we have some lines. We call them again barrier, mechanical barrier defense. We call them again a first line of defense. Under first line of immune response, under innate, we can have some anatomic sites. Yeah, or body substances like a skin. Skin may defend us as in epidermal surfaces. Skin also, under skin, we have what you call a sweat or secretion. And oral cavity also contains some enzymes called lysozymes. Stomach, you know, that contains some it's acidity environment. Gastrointestinal tract again may have some low pH. Mucosal surface, you know, mucosal surfaces, epithelial cells. Those are the part of the that contribute as a mechanical barrier. So it just prevent prevent an entering invasion of a microorganism or any antigen. Normal flora, you know, normal flora in bacteriology are those normal flora bacteria which cannot develop a disease in the human body. They have some kind of roles in preventing other pathogenic organisms. If you remember the function or the role of normal flora bacteria. Yeah, that is it. You can see now some saliva in your mouth, the acidity in the stomach, the mucus layer in the gastrointestinal tract, all of them, the ciliated epithelial cells in the respiratory tract, all of them, they contribute as a first line of defense of the immune system. Next. I want now to let you know a second line of defense after a first line or mechanical barrier. We can now have the role of immune cells. This is all under innate immunity. We have a mechanical barrier or anatomic site and the body substances. So next we are gonna now to talk about the role of cells of innate immune system. So for this now, we can now talk about under these innate immune cells, we can now have two parts. We have this area of antigen-presenting cells. The antigen-presenting cells, we can now mention, we have a B cells. B cell is not part of innate. These B cells can play a role in presenting antigen. B cells are considered adaptive immune cells, but have an extra function of antigen-presenting cells. We have also a macrophage and the dendritic cells. These three, they are part of antigen-presenting cells, but these macrophage and the dendritic cells are a part of innate immune cells. And we have another group of innate immune cells that contribute in killing antigen. One, it is natural killer cells. It plays a major role in destroying infected and transformed host cells, major role in viral infection and cancer immunity. Are you following here? Actually, natural killer cells is a lymphocyte cells, but their function is under nanospecific response. Can you see natural killer cell here? It can now destroy abnormal host cells. Also, they can now fight against the cancer, the cancer cells. Sometimes they can now against a viral infection. We have now what we call we have a mast cells. Mast cells, and this is a role is the allergic response and even a mucosal surfaces. So under now innate immune cells, we have what we call a polymolecular leukocyte. Means they may contain more nucleus and even also some granules. We can now have an eosinophil and a basophil. You see basophil? A basophil and the eosinophil, these eosinophil and the play a major role in the allergic response and the parasitic infections. These release histamine, leukotriene, heparin, and other toxic products. Toxic parasites and they cause damage to the hosts, and this one, especially in allergic and anaphylactic response. Make sure that understand this one, eosinophil and the basophil. This one are both intervene in allergic and the parasitic infection. This neutrophil, actually neutrophil, it's is circulating in blood. Then first to encounter pathogen, major role in fungi and the bacteria, even the viral infections. That's why you people, when you have a bacteria infection, fungi, and even also some viruses, you can see elevated of neutrophil in the blood cells. Yeah, that's it. So meaning of these antigen presenting cells, B cell, macro and the dendritic cells, are those one that encounter first with the antigen and they present the antigen to helper cells. So it connects the immune cells, it connects the innate and the adaptive. So we will explain more for the next slide. The adaptive immunity now, what is it? I've told you that there are specific immune response. This refers to a third line, means the third line of defense. Acquired specific immunity is the product of a dual system of a unique leukocyte, B and a T lymphocyte. Two features that most characterize adaptive immunity are specificity and memory. You have to note this. This adaptive must be characterized, this specificity in terms of action, in terms of function. They are more specific. Number two, they have this characteristic of keeping memory of the second exposure or further exposure in the future. So these, the antibody produced against the chickenpox viruses can't fight against measles viruses. So each infection have a specific response. That meaning of specificity. Memory, the property of memory patterns to the rapid immobilization of lymphocytes to have been programmed, recall their first engagement with the invader and they respond rapidly during subsequent exposure. Yeah, and then what you have to know for this adaptive is now the function of T lymphocyte and the B lymphocyte. T lymphocyte and the B lymphocyte are those one governing what? Adaptive immune response. Next. Yeah, you can now imagine how what composed adaptive immune cells. The adaptive immune cell is broken into two parts. We have what we call cellular immunity and immunoimmunity. Cellular immunity means what? It's mediated by T cell. So it is the function of the cell itself. So cells produce cytokine, cell produce protein to kill an antigen. So we have immunoimmunity. Immunoimmunity means what? Is mediated by B cell. What B cells do or does, it's produce antibodies. Do you know antibodies? It produced antibodies. So immunoimmunity refers to a function of antibodies. Cellular immunity refers to the action of cellular itself by producing some protein called cytokine. Now, if you go to this cell mediated by these T cells, you can see now T cell is composed by CD8. CD8, T cytotoxic T cell. This is recognized by MHC1. And then this CD4 helper T cells. Actually, a T cell have subtype. A subtypes of CD4 and CD8. Okay? And the CD4, it's what we call a helper T cell. We can call them as a chief in command of all immune system. So a part of this, CD8, their main function induce apoptosis in the target cells. So you can see when you have some abnormal cells or infected cells by cancer or a viral infected. So this CD8 all kind of intracellular infections. This CD8, once it is activated, it can now kill and destroy all these abnormal cells. Now, if you talk about helper T cell now, it have what we call subtype, depending on the type of infection. These T helper cells is having a CD4 helper called T helper 1, T helper 2, and the T helper 17. Okay? A T helper 1, once it is now activated to differentiate into a T helper 1, this will be activated, macrophage to destroy pathogens, and also will activate B cell to produce an antibodies. Okay. Now T helper 2, their role, it interacts with B cell mainly for the purpose of antibody production. So T helper 17, in the case of fungi infection, it's where T helper 17, it will now lead this kind of infection. I hope that you are following me and understand this process, okay? So CD4, it differentiates, it's activated based on the type of infection. If it is a fungal, T helper 17 will be responsible to handle the problem. When in the case of antibody production, either T helper 2 will be responsible, even also T helper 1 in the case of antibody production, and also macrophage activation. Okay. So if we move on immunoimmunity here, you can see B cells. B cells, once it is active, normally B cells remain in your blood as inactivated form. Once it's activated, it transforms into a mature plasma cells. Plasma cells will now secrete antibodies. You know the antibodies? You can see we have around 5 antibodies. We have IgM, we have IgD, we have IgG, we have IgA, and the IgE. You see, we have around 1, 2, 3, 4, 5 antibodies. And each antibody have a specific function. And above all, this T cell can also, okay, can also keep the memory. Even the B cells produce plasma cells that directly secreted, okay, by the antibody. So these B cells and the T cells must turn into a memory cells for the next infection. I think I will try to explain this this slide. Next. So after understanding now what the component and the function of innate and adaptive, you need to understand what is immunization now. Okay, immunization is to make your body strong. So immunization is defined as a process by a person is made resistant to a disease, typically by the administration of a vaccine. We know that the purpose of vaccine just is to make a person to be strong to fight against many diseases. Yes. So this immunization now, the body must create 2 types of immunity. One, it is immunoimmunity and the cell-mediated immunity, you remember. So the immunization, immunoimmunity must be strong. Even cell-mediated must be strong. to have a strong protective mechanism. So immuno are those mediated by antibodies in various body fluids, including blood, saliva, and the mucus secretion of the lungs and intestinal tract. In general, the immune response offer protection from infections caused by organisms which are extracellular, and a category which include most bacteria as well as many of the toxic products, even hay fever and food allergies. Note that immunity is more efficient in extracellular infection, such as bacteria extracellular or toxic chemicals, food allergies, food intoxication. So those microorganisms which cause a disease while they are extracellular, this immunity is efficient to fight with those kind of infection. Okay. So a cellular immunity, it's called cell-mediated immunity because it of protective factors are living cells. Cell-mediated immunity is responsible in general for resistance to infectious organisms, which are the primary intracellular. These include resistance to the viral infection, as well as certain bacteria, mycobacteria, even some intracellular fungal, even also graft rejection. You understand, my student? So you have to know that these cell-mediated, they are targeting all kind of intracellular infection. You remember that all viruses are obligate intracellular infection. Some bacteria also, they are intracellular, like Mycobacterium tuberculosis, like chlamydia, like rickettsia. All of them, even some fungal, which are intracellular, these cell-mediated are responsible to eliminate those intracellular infections. Even also those graft rejection during a transplantation mechanism, rejection of a graft or a kind of cancer cells, those abnormal cells, cell-mediated, is responsible to eliminate or to fight against of those group of infection or those group of disease. Okay, I think it's clear now. A general system for classifying immune system, you can see we have a person can now acquire several immunization, okay, or several immunity. We have what we call natural and artificial. Natural means a passive, the way it's a mother to the fetus or mother to babies during breastfeeding. Yeah, that is how we call a passive maternal immunization. We have again active infection. So during the immunity produced after infection. You are infected with the microorganism, your body creates immune responses even to protect you for further infection. That is a natural. It doesn't need any vaccine, so your body creates itself the immunity against infection. So under artificial now, some immunity is prepared or manufactured out of your body in the laboratory or industry. So under now artificial, we have a passive antibody transfer or active immunization. Passive means You know, what we call immunotherapy. You know immunotherapy. So a scientist prepare the antibody in the laboratory. So during now, maybe you may have maybe immunodeficiency, you may have a weak immune system. They inject you already prepared immune cells. Okay. So what we call immunotherapy. They inject you antibody, they inject you immune cells, depend to what your body need. So active means they create the artificial immunization from the vaccine. So they inject you the vaccine for the purpose of the specific microorganism. So it's artificial active immunization. So that's how we can now classify a group of immunity people acquire during their lifetime. Okay. Now, you need to know, you need to know, you guys, how this innate and adaptive connect each other, how they work together. So there is what we call antigen presenting cells. Antigen presenting cells, it connects innate to adaptive immunity. Remember that adaptive is the third line of immune response. Okay, sorry. So remember again, the antigen presenting cells in the first slide I've told you that it's B cell, macrophage, and the dendritic cells. You have to note that antigen presenting cells are those of B, macrophage, and the dendritic cells. These cells, they used to connect an antigen to helper cells. So this is the antigen presenting cells. A bacterium or an antigen is engulfed by a macrophage, is encased in a vacuole. Lysozymes fuse with the vacuole and digest the bacteria within the cell, the vacuole. You see this process, bacteria there, internalize within the vacuole of a bacteria. So we have a molecule called major histocompatibility complex. It's called MHC2. These molecules, it always bind with this peptide antigen within the cytoplasm. Then this MHC, it expresses on the surface of these antigen presenting cells. The antigen from these, from digested bacteria, are represented with the MHC class 2 on the cell surfaces. Do you are following me? Now, the antigen presenting, such as the macrophage, engulf a foreign antigen, partially digest in the lysosomes, and then embed in the MHC class 2 molecules for presentation at the cell surfaces. Lymphocytes, remember lymphocytes is representing what? Helper cells, representing what? Adaptive immune response. Lymphocyte of adaptive immune response must interact. You know how adaptive interact with the antigen? It interacts with the antigen embedded MHC class 2 molecules to mature into functional immune cells. Let me now, we have two kinds of major histocompatibility complex called MHC. We have MHC1 and MHC2. So means the lymphocytes as the adaptive immune response interact or recognize the antigen embedded MHC class 2 molecules to be functional immune cells. Okay. Yep. I want now to let you know what you call antibody. after now lymphocytes recognize the antigen, immune system may prefer to produce antibodies. So what is it, antibody now? The antibody structure looks like this. It have around 2 chains. We have a heavy chain and a light chain. Do you see the light chain? We have a 2, 1 and a 2. And the heavy chain, it's long. It's longer than light chain. You can see. And then each chain contains 2 parts. We have a constant region and a variable region. You know, variable region are those in pink or red color. You see red color? Red color is presenting a variable region of a heavy chain and the variable region of what? A light chain. Okay. So you know the role of this variable region. Do you see here? This variable region, it's antigen binding site. It's where antigen binding with these antibodies. Once the antigen bind with this antigen on variable region, they have what we call a signaling. The signals will pass through this constant region, because this constant region here, it always binds with the cells. This constant region, it's bind with the cell surfaces of all the immune cells. So there is somewhat we call a communication. Some signals from this binding site to constant region up to internal cells. So the cells will sense a signal from a variable region, pass it through the constant region up to cellular area. Okay? Yeah, I think this is clear now. So you can see we have a heavy chain from the red one up to C terminus. Yeah, we have a light chain, is a small, and the heavy chain, the long ones. Okay, and they have a shape of a Y shape. Good. So we have around 5 antibodies, and each antibody have a specific function. Let's start by IgA. Actually, IgA used to be under mucosal area. Okay. In the mucosal immunity, it's the first antibody to encounter with any types of microorganism. We have IgD. It's naive, B antigen receptors. Okay. It's always binded. It's binded of B cells. It can be considered as a B cell receptor. You know receptor. Receptor, it first encounter or attach to an antigen. It's a bounded antibody on B cells. IgE. IgE actually it's a monomer. It's defense against helminthic parasite, immediate hypersensitivity. You see, IgE it's very high in worms parasites and also allergic condition. IgG, you know, IgG, it's opsonization, complement activation, antibody-dependent cell-mediated cytotoxics, and the neonatal immunity and the feedback inhibition of B cells have a great job in this immunoimmunity, IgG. Even this one can persist longer in your blood, in your serum. Okay. It can also protect your babies in the fetus in the womb. Okay. So it can pass it through your placenta and they protect your baby. Now, IgM, it's also a bounded immunoglobulin on the surface of a B cell. Okay. It's naive B antigen receptors if I'm asking you questions, we have a 2 immunoglobulin always bounded on B cells, serve as a receptor of B cells. Those immunoglobulin or antibody, they are IgM and IgD. This IgD and the IgM, they are a bounded antibody on the surface of B cells. They serve as a receptor of B cells, right? Okay. So now I want to let you know a brief summary of the main function of antibodies. Once antibodies are released or are produced, they have a function of neutralization. You know what kind of neutralize? Neutralization means antibody prevent a virus or a toxic protein from binding to the target cells. Do you understand, my brother or sister? Okay. Neutralization is a kind of preventing this antigen to bind to a specific receptor of a cell. Okay. So it can now, you know, sometimes some viruses may remain out of the cells. So this kind of antibody prevent, even stop this virus to attach to a specific cell target. Okay. Opsonization as the second function, opsonization. A pathogen target by an antibody is consumed by a macrophage or neutrophil. So you see, complement activation. So actually, even the antibody have this capability of activating the complement cascade, the complement system. There are protein found in the plasma, So they serve in immune responses, in innate immune system. So these are the main function of antibodies, but we have many, but these are the main function of antibodies. That's how during HIV, the antibodies can be able to neutralize the viruses before entering the cell, leading to destruction or to phagocytosis by the macrophage and lead to elimination. Great. Yeah. Let's now talk about, those are the three, there's a basic skills and the knowledge of what we can know about basic fundamentals of immune system and their function, yeah, even their component. So now I want to let you know what we call disorder of immune system. Actually, three main functions of immune system. It can remove infectious pathogen. It can kill cancer cells. It can also contribute in tissue repair. That's the main function of immune system. Okay. Now, sometimes immune system may be deficient. You know deficiency? They are not enough. They are weak. These, once they are deficient, response may lead to chronic infection or a cancer. So this refers when your immune system is not enough. They are deficiency. Number two, sometimes immune system may be excessive response. It's referred to overreacting or inappropriate response. Okay. These inappropriate responses of immune system or excessive response may lead to hypersensitivity disorder Okay. Third one, sometimes your immune cells, they are self-reactive. So means it reacts against a self protein or against with the own cells. So that is also a problem. These immune functions may lead to some disease or disorder in 3 ways: chronic infection, hypersensitivity, or autoimmune. The first one, chronic, this one is called immunodeficiency. Yeah, so those are the 3 disorders of immune system. Next slide. Yeah, let's start by immunodeficiency disorder. How? Immunodeficiency is a state in which the ability of the immune system is compromised or entirely absent to fight against infectious disease or a cancer. Okay, the 2 types of immunodeficiency disorder, it can now happen in 2 ways: primary or congenital or inherited disorder. Okay. Some commonly variable, some example, we have a commonly variable immunodeficiency, we have a DDoD syndrome, we have a severe combined immunodeficiency. Those are the types of disease or disorder that you may acquire during congenital or inherited. It means it's parental transmissions. It's hereditary. You're born with. You cannot prevent this primary. Huh? It's hereditary. Number 2, some immunodeficiency, these states, you may acquire after birth, what we call secondary or acquired immunodeficiency. This disorder happens when an outside source like a toxic chemical infection attacks your body or immune system. For example, some severe burns, burns may lead to insufficient immune system. Chemotherapy, most of the time they are a cancer drug. The cancer drug, a prolonged cancer drug may lead to immunodeficiency. Some also radiation. Radiation may target one types of immune cell and they lead to immunodeficiency. That's why you guys, a multiple exposure with this X-ray, even also some radiation, may lead to some destruction of or killing of some immune cells. Chronic disorders, chronic disorders like such as diabetes, even some cancer, and so on. This can lead to immunodeficiency, like a cancer, malnutrition. I cannot also forget what we call also HIV. HIV disease may lead to immunodeficiency. You have to remember also AIDS as one of the cause of a secondary immunodeficiency. Yeah. Number 2, disorder of the immune system, hypersensitivity disorder. Remember that this is referred to excessive or inappropriate immune response. So we have a type 1 hypersensitivity. This refers to allergy disorder or allergy disease. Some people complaining, I have allergy, respiratory allergy, rhinitis, everything, skin allergy. So this reaction now, it's what we call hypersensitivity reaction. Okay. So hypersensitivity type 1, it's called again allergy, is where the body reacts to something that normally harmless, like a pollen, pollen, dust, or animal form. So the symptom can be mild or a serious. This kind of allergen. We have some allergen means the antigen cause allergy. Allergy is a disease, and antigen is called allergen. Some common allergen, we can list some of them. We have a food, peanuts, fish, shellfish, milk, soy. So you see, you can hear some people say, I have an allergy of milk, allergy of what? Some food, and so on. We have some people also, they have allergy during insect bites. When insect bite you, you can see some skin reaction, even some symptom in your skin. Pollen, when some people walk around in the garden and inhale some pollen, okay, or animal dander, house dust mite. So these people, they are now allergen of respiratory cavities. Some people may have a rhinitis, okay, and so on. Drug also. Remember always, some drug injection may lead to allergy, like a penicillin. That's why they told you, don't inject a penicillin without antidote apart, okay, to stop the reaction. Penicillin, even some cosmetics, some cosmetics chemicals may lead to skin allergies. You can see now some symptoms in these pictures. You can see systemic skin infection, skin rashes. You can see now respiratory tract symptom. These children. So those are the types of allergies, and this you can see the cause of these allergies. Okay. Next. Next slide. So while waiting the next slide, no, actually this hypersensitivity, the body must sensitize the antibody for the first encounter. So these allergens happen for the second and the third exposure because the immune system may already develop this antibody on the first infections. Okay, next slide. Yeah, okay, no problem. So a type 2 hypersensitivity disorder, it's happening when the IgG or IgM antibody bind to the cell surface antigen and activate the complement system, leading to cell damage, phagocytosis, or antibody-dependent cell cytotoxic by natural killer cells. Do you know why what we call blood group incompatibility? That's what when they say a B cell, a blood group B cannot give a person who have a group B, group A. This is due to hypersensitivity type 2. Because if you receive an incompatibility blood, the antibody of a receiver, okay, will now target the blood group antigen on the cell surfaces of the blood of donor. This antibody antigen interaction happening on the cell surface of the blood cell or red blood cells will lead to inflammations. And this inflammation now will lead to destruction of red blood cells of a receiver. Okay, so means a recipient who receive the blood donor or the blood from a donor, the antibody from a recipient will target the blood group antigen. And this reaction, antibody antigen on the cell surface of red blood cells will activate the complement system, also lead to phagocytosis. And this reaction would lead to what we call hemolysis. Hemolysis means excessive destruction of red blood cells due to these hypersensitivity reactions. Why it's called excessive or overreacting? This reaction may require few hours to react and cause hemolysis. This reaction happens in the case of a transfusion reaction, erythroblast erythroblastosis fetalis, autoimmune hemolytic anemia, even some drug reaction. So these are the mechanisms of immune system, okay, leading to a disorder. Yeah, next slide. Now, type 3 hypersensitivity, it's when the antigen combine with the antibody in circulations, blood circulations, and form an immune complex, which is deposited in an organ. Immune complexes activate the complement ecosystem, leading to recruitment of polymononuclear leukocytes, result in inflammation and the tissue injury. Any question there? Or can I move? Can I move? Okay. You see, hypersensitivity type 3, it's happening when the antibodies of a human body encounter with any antigen in blood. The antigen antibody form what we call antibody antigen complex molecules. These molecules will accumulate, okay? These immune complex may accumulate and deposited in different organs. This deposition now will recruit or activate the neutrophil, even some white blood cells. This will result in inflammatory reaction. And inflammation, you know, it's all about sign and the symptom. And they will cause some tissue injury. Okay. Now, you can see some of the examples of hypersensitivity type 3. Rheumatoid arthritis. Immune complex are deposited in the joint. You know joints? Results in release of inflammatory mediators. And the joint break down and they become distorted. Treated with anti-inflammatory drug. Are you still with me? Raise your hand. I want to see if you are still with me. Are we on the same page? Thank you. So this kind of inflammation may lead to joint disease. See, that's how this is happen when just an immune complex are deposited in the joint. Then this joint will now break, will be break down and they become distorted due to inflammatory reaction that is happening in the joint. Okay. Yeah, we have again a type 4 hypersensitivity. This is a kind of reaction involves T cell mediated reaction. T cell or macrophage are activated and result in the production of a cytokine which induce tissue damage. An example now, this hypersensitivity happen in the case of a contact of a poison. You know poison? Reaction to metals of jewelry, tuberculin type hypersensitivity, granulomatous hypersensitivity, leprosy, even also some schistosomiasis. So this kind of reaction mediated by T cell and the macrophage may result in a serious reaction. Okay. Okay. So autoimmune disorder, you know autoimmune, I've said that these autoimmune happen when the body natural defense system can't differentiate the own cell and the foreign cells, causing the antibody to mistakenly attack normal cells. Can you imagine how your immune cells attack the own cell? Or your immune system attack one of your protein. Imagine now, it's what we call autoreactivity, autoreaction. So examples. Can you imagine now the type 1 diabetes mellitus? Type 1 diabetes mellitus. It's caused by autoimmune reaction. It's caused by auto attack against insulin producing cell, beta cell, the langeron of the pancreas. This results in a decreased production of insulin and a rise in blood glucose level. So actually, this type 1 diabetes you cannot prevent, and it's a chronic. Some people born with it's a young diabetes because you used to have some immune system always attack the beta cell of the pancreas producing insulin and glucagon. You know the role of insulin? It's raise the blood sugar levels in the blood and the reduce, let me say, insulin reduce the blood glucose levels. A glucagon increase the blood sugar levels. So the insulin now here in the diabetes, it's affected, is no longer now produced because the cell has been now damaged by immune reactions. Number 2 examples of disorder of under autoimmune disease. We have systemic lupus erythematosus. This is when autoimmune in this disease, the immune system of the body mistakenly attack healthy tissue. It can affect skin, joint, kidney, brain, and other organs. So these are the one of some disorder. There are many. There are many. So those are the disorder. You can now expand, search yourself, and read more about those immunological disorders. Okay. So now, at the end in immunology course, it helps us to know how to diagnose some diseases. We use immune system to diagnose a disease. Okay. So some immunological laboratory now we use in the lab, we can now use agglutination reaction, precipitation reaction, enzyme-linked immunosorbent assay, ELISA. We have a flow cytometry, and the detection of lymphocyte subgroup, functional test of lymphocyte, measurement of phagocytosis, a phagocyte. Yep. So this is it shows how the antibodies here react with the antigen, the viral antigen, and you can see these green ones. It is the indicator. Once there is a positive reaction between the antibodies, here we have indirect. Indirect means we have two antibody together. One carry what we call indicator, phenol, and one antibody coat with the antigen. So once there is a presence of antigen in the sample, there is a presence of a color change in the reaction. Here we have one single antibody interact one single antigen. If there is a presence of antigen in the sample, with the presence of this indicator, the positive reaction will appear as the color changes. So this color change show a positive or a presence of an antigen in the samples. So you can see now the immunological reaction may help us to diagnose the presence of the antigen in specimen. Yeah, we have another kind of indirect fluorescence antibodies. Those are the different technique used in the laboratory. So thank you very much for your kind attention. Thank you. We are done. We are done. Thank you very much. Any question there? Do you have a question?
Okay, so thank you, Jeff.